Hi everyone, my name is Leon. I'm currently a PhD student at the University of Toronto. Today, I'm here to talk to you about how deep learning enables automated and extensible peak group identification for multi-transition chromatogram-based data-independent acquisition data analysis. What a mouthful. So what does that mean? Well, data-independent acquisition, or DIA, is a mass spectrometry method which aims to be more reproducible and quantitative than traditional shotgun mass spectrometry methods. Instead of stochastically sampling precursors to further fragment, DIA uses deterministic peptide ion isolation windows instead, shown over here. And from the resulting data matrix, we can actually extract time series-like signals called chromatograms. The resulting data matrix itself from DIA methods can be very multiplexed and contain large amounts of information. So that's why we need scalable and sophisticated software tools to deconvolute this data before it can be used for further analysis. So current methods like OpenSWAP coupled to PyProfit operate by applying heuristics to propose peak group candidates, and then using manual feature engineering to convert those peak group candidates to a series of scores that are passed into a traditional machine learning classifier, like a linear classifier or XGBoost, to be given an aggregate peakness score. This stage-by-stage pipeline can lead to errors occurring at each stage that are propagated downstream. My goal is to create an automated peak group detection method that can take full advantage of the richness of DIA data and directly without as many intermediate steps. So to do this, I adapted a semi-supervised framework for deep learning, originally for image classification, called FixMatch. And I used it to train deep learning models that operate directly on chromatograms instead of peak group feature scores. Due to the fact that mass spectrometry produces more data that can be annotated by people, to truly take advantage of the data, you must be able to use unlabeled data in addition to labeled data. So I trained the model on a Streptococcus dataset containing approximately 5,000 manually labeled chromatograms, 90,000 decoys used as true negatives, and approximately 220,000 unlabeled non-decoy chromatograms, which consisted of roughly 70% of all available chromatograms in proportion. And the results are as follows. Comparing the semi-supervised neural network to a series of baselines on two metrics. The first metric is simply the ratio of targets called to the number of decoys identified, creating a pseudo false discovery rate to recall curve. Here we compare a purely supervised neural network trained on the 5,000 manually labeled chromatograms in the training set, shown here on the red curve, an XGBoost-based PyProfit model where the spectral features like mass accuracy and isotopic envelope overlap have been removed because those features are lost when you're converting to chromatograms, so the neural networks don't have access to them. And we also compare it to a version of the PyProfit with all the features without any of them taken out. So we can see from these two graphs that the semi-supervised neural network is superior to the supervised network and comparable to PyProfit on equivalent data. However, both the PyProfit with no special features and a semi-supervised neural network show a performance gap between the PyProfit using all the features. And it just goes to show how important those special features are to the final classification performance. The second metric is a traditional precision recall curve on the holdout manually labeled data. We would compare a supervised XGBoost model, shown here in purple, against the XGBoost PyProfit trained on all features in green, as well as our semi-supervised neural network, shown here in red. The semi-supervised neural network is able to perform on par with supervised XGBoost, whereas PyProfit fails to keep up. So from these results, it seems that both supervised and semi-supervised models generally see a trade-off between the two metrics, where supervised models perform better on the true labels, but semi-supervised methods hold the edge on pure decoy false discovery rate. But the semi-supervised neural network is, at least seems to be robust on both, at least for this data set. Finally, we also draw some samples from the holdout test data. And we see that the neural network produces qualitatively superior peak boundaries, shown here in red, compared to the open swath proposals in blue. I'll zoom in a little bit more. The neural network, it seems, is able to learn the nuances of the data from the manual labels to produce more plausibly human-like peak annotations. You can see in the examples here, generally the open swath labels are too narrow or too wide, but the neural network proposed peak boundaries are probably closer to what a person looking at these pictures would select as the annotation. So in conclusion, it seems like deep neural networks are definitely capable of learning salient features directly from chromatographic inputs. They perform favorably and robustly compared to existing baseline methods. And they also seem to have promising qualities for better quantitation. In the future, I aim to use inputs derived directly from the raw data to incorporate more information to push performance beyond current state-of-the-art, implement weak label training to further decouple the model from human intervention, and to incorporate information from across runs and experiments to take full advantage of the highly structured nature of DIA data. Thanks for watching my presentation.